Hello, biologists. Let's finish out our discussion of Chapter 2 by talking about DNA and RNA, kind of the king of your cell. These are called nucleic acids because they are found in the nucleus. And for a long time, science just didn't know what they were doing. We just knew they were in the nucleus. So they were called nucleic acids. Now we know they are storing and processing information. All the information your cell has about what color eyes you should have, what color hair you should have, if you're able to digest lactose, if you're going to make freckles, how your fingernails grow. All of that information comes from your DNA. So your DNA is really the blueprint that has all the rules that will determine what characteristics you've inherited from your parents. The DNA you have is a copy of DNA from your mother and your father. And when you have children, you pass that on to them. And when they have children, they pass it on to your grandkids. DNA does its job by directing protein synthesis. We'll have a lot of fun in Chapter 8 when we start discussing how it does that. But it has all the rules for how to make proteins. So it's going to control what enzymes are being made. And by controlling what enzymes are being made, your DNA really is in charge of all the metabolism, everything that's going on in the cell. However, we know DNA stays in the nucleus. So it is going to make kind of a middleman that's going to leave the nucleus and go out into the cell to actually moderate those intermediate steps in making proteins and all of that work. And that's called RNA. So DNA is what you inherited. It stays in the nucleus. RNA does the intermediate steps to make proteins. It is made in the nucleus, but then goes out into the cytoplasm to do its work. DNA and RNA both have a monomer called nucleotides. So in this pearl necklace of nucleic acids, the monomer is a nucleotide. Nucleotides are made of three things. One is a sugar. There's one of two types of sugars that could be here. Deoxyribose if it's DNA, and ribose if it's RNA. So the difference between deoxyribose and ribose is you have lost an oxygen. So D means lost an oxygen. So deoxyribose or ribose for DNA or RNA. There's a phosphate group. Remember talking about that PO4 group when we discussed functional groups? That shows up here as a phosphate group. And a nitrogenous base. There are a few different bases it could be. It could be A, G, T, C, or U. So where we had 20 amino acids as our monomer for proteins, here we have 5 nucleotides, 5 nitrogenous bases as your monomer for nucleic acids. These nucleotides can be divided based on their shape. Some are bigger and some are smaller. The bigger ones are adenine and guanine, and they're called purines. The smaller ones are cytosine, thymine, and uracil, and they're called pyrimidines. So I remember the bigger molecule has a smaller name, smaller molecule has the bigger name. Adenine, guanine, and cytosine, all three of these are found in DNA and RNA. However, thymine is only found in DNA, and uracil is only found in RNA. So it just changes a little bit of a functional group here for this to be in DNA, this to be in RNA. When we compare DNA and RNA, although they both are very similar, there are some very important changes between these two. So DNA is double-stranded. RNA is usually going to be single-stranded. So DNA is usually made of 2 strings that are held together with hydrogen bonds between them, while RNA is just 1 strand by itself. DNA is going to usually be found as a twisting double helix. It looks like a spiral staircase, just twisting around, where RNA is going to have different shapes. In DNA, you'll have a purine pair with a pyrimidine, so a small with a big, adenine with thymine, cytosine with guanine, and that allows those 2 strands to, if one side has a smaller and one side has a larger, the entire width is going to be consistent. So the width of your spiral staircase, the width of your double helix, is always going to be consistent, whether you have adenine and thymine together or cytosine and guanine together. RNA, because it's usually single-stranded, we don't see so much of that hydrogen bonding between two strands because it's one strand. RNA also comes in 3 different forms: messenger RNA, transfer RNA, and ribosomal RNA. You can also abbreviate these mRNA, tRNA, and rRNA. So here, if you look at DNA, you can see this twisting spiral staircase going up, where RNA is just a straight thing. You can see G and C bonding together. Anywhere you see a C on one, you see a G on the other strand. And you see A and T bonding together. Anywhere there's an A on one, there's a T on the other. A little mnemonic to remember how they bond is apples are in the tree, cars are in the garage. So apples in the tree, A and T, cars in the garage, C and G. You don't see a lot of that hydrogen bonding here because there's just not another strand to bond with. And this allows the smaller pyrimidine and the larger purine have a constant width. So this is this wide. The larger purine, the smaller pyrimidine, have a constant width. So this DNA, the distance between the two strands, stays the same no matter what base is there. You can also tell RNA is smaller than DNA. DNA could have hundreds of thousands of bases. RNA will have, maybe hundreds of bases. So if we compare them, RNA has ribose, DNA has deoxyribose. They both have a 5-carbon sugar, it's just different. RNA has A, G, C, and U. DNA has A, G, C, and T. RNA could be fewer than 100 nucleotides up to tens of thousands. DNA is going to be millions of nucleotides. The shape of RNA is going to change depending on what kind it is. If it's mRNA, rRNA, or tRNA, it has variable shapes. But DNA will be 2 strands coiled together in a twisting double helix. RNA does protein synthesis as directed by DNA. DNA stores that genetic information. It keeps the information to control protein synthesis. DNA stays in the nucleus, whereas RNA can leave the nucleus to go into the cytoplasm to direct those steps of protein synthesis. Nucleotides are also the basis of our high energy compounds. These are compounds that our cells use to store energy. So if you were to eat something, you don't just have to immediately burn that energy off or lose it. You can store that in this molecule called ATP. ATP stands for adenosine triphosphate. That means it's an adenosine with 3 phosphate groups on it. If you break off that phosphate group, if you break off one of those phosphate groups, you release energy and you're left with ADP, or adenosine diphosphate, with 2 phosphate groups. And then if you eat something again, you take the energy from your food to put that phosphate group back on to have ATP. So you can phosphorylate ADP. Phosphorylation is adding a high energy phosphate, or you can dephosphorylate when you take off a high energy phosphate group. I do want you to mark this out though. The enzyme that does this is ATP synthase. ATP synthase catalyzes the phosphorylation or addition of high energy phosphate groups. ATPase will synthesize dephosphorylation. ATPase catalyzes dephosphorylation. So ATP synthase turns ADP into ATP. ATPase turns ATP into ADP. So here's a look at ATP, adenine and ribose, and 3 phosphate groups. Just like if you had 3 kids in the back seat going on a long car ride, they have lots of energy. If you only had 2 kids, if you lost 1 kid, you just lost some of that energy. So by putting these phosphate groups together, you have a lot of energy between them. So they're really good at storing energy. So you can break this phosphate off and release energy. You can break this phosphate off and release a little bit of energy. But your cell is constantly cycling between breaking off phosphate when it needs to release energy and putting on phosphate when it needs to store energy. Okay, so at this point you can tell me, what are the monomer of nucleic acids? How do DNA and RNA differ? And how are they similar? And which of these provide more energy for a cell and why, ATP or ADP? Okay, we have now finished your first 2 chapters. You're halfway to your first exam. If you have any questions, please don't hesitate. This is a time to come in and clarify things so that you can stay on track for this whole semester, get off to a good start. I look forward to seeing you soon, and we'll be picking up with chapter 3 in our lecture discussing microscopes. We're getting into the fun stuff next. Bye.